In this discussion, we're going to be talking about Chapter 2, Atoms, Ions, and Molecules. Matter is anything that has mass and takes up space. In the body, matter exists as solids, such as bones and organs, liquids, such as blood and lymph, and gas, such as oxygen and carbon dioxide. An atom is the smallest unit of an element with chemical properties. Pure substance. An element is a pure substance made up of one type of atom. There are 92 naturally occurring elements. Hydrogen is the smallest and lightest, and uranium is the largest and heaviest. The periodic table of elements is a chart that organizes all known elements and is used to identify and study elements in the body. In the body, there are major elements that make up 99% of body weight, minor elements that is less than 1%, and trace elements that is less than 0.01%. The 12 elements found in the body above trace levels include major elements of oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus, and minor elements, sulfur, potassium, sodium, chloride, and magnesium, and iron. An atom is broken into subatomic particles. Protons have a mass of 1 atomic mass unit, and the charge is positive, +1. Neutrons have a mass of 1 atomic mass unit, and the charge is 0 or neutral. And electrons have a very small mass charge of -1 and are found in orbitals around the nucleus. Atomic mass unit is a standard unit for measuring atomic mass, also known as Dalton. The nucleus is the center of the atom and contains the protons and neutrons and holds almost all the atom's mass. The orbitals are areas around the nucleus where electrons are located. A chemical symbol is 1 or 2 letter abbreviation for each element. It's based on the element's name, often in English or Latin. An example would be H for hydrogen or Na for sodium. The atomic number is the number of protons in an atom. It is found above the symbol on the periodic table. Elements are arranged in order by atomic number. Atomic mass is the total mass of protons plus neutrons and represents the heaviest of an atom. This is used to compare the weight of different elements. Protons, the number of protons are equal to the atomic number. Electrons are the same as protons in a neutral atom. Atoms are neutral, so the number of electrons is equal to the number of protons. Neutrons is equal to the atomic mass minus the atomic number. So mass equals protons plus neutrons. This book uses the shell model. The shell model is where electrons move in energy levels known as shells around the nucleus. Each shell has a specific energy level. Electron capacity by shell is within the first shell up to 2 electrons, the second shell up to 8 electrons, and the third shell and beyond at least 8 electrons. The filling order, the inner shells fill first before electrons move to outer shells. This helps determine how many atoms bond and interact. Isotopes are atoms of the same element. They have the same number of protons and electrons, but different number of neutrons. They have the same chemical behavior, but different atomic mass. An example is the carbon isotope with carbon-12, carbon-13, and carbon-14. The average atomic mass of carbon is equal to 12.01 atomic mass unit. Average atomic mass is the weighted average of naturally occurring isotopes. Radioisotopes are unstable isotopes with excess neutrons and emit radiation as they decay or break down to become stable. Physical half-life is the time for 50% of radioisotopes to decay or break down, and biological half-life is the time for 50% of radioisotopes to be eliminated from the body. A valence shell is the outer electron shell of an atom. The number of electrons in a valence shell determines reactivity. Elements in the same column have the same number of valence electrons. Atoms are chemically stable when their outer shell is full. Usually this means 8 electrons in the outer shell. Noble gases, or the group of elements in column V8A, have a full valence shell. They do not form bonds easily and are chemically stable. The octet rule is where atoms gain, lose, or share electrons to reach 8 in the valence shell. The goal is to achieve chemical stability. A covalent bond is formed when 2 atoms share electrons. That will be discussed further in a bit. An ion is an atom or group of atoms with a positive or negative electrical charge. This is formed by losing or gaining electrons. Cations are positive ions that are formed when electrons are lost, and an anion is a negative ion formed when electrons are gained. I would also recommend reading the sections in the book that are listed below. Ionic bonds are formed when positively charged cations and negatively charged anions are attracted to each other. The attraction is due to electrostatic interactions between opposite charges. Salt is the structure that results when a cation and anion combine. Typically refers to ionic compounds formed by these ionic bonds. An ionic compound is a substance made up of oppositely charged ions. These ions are held together in a lattice crystal structure by ionic bonds. The lattice arrangement maximizes attraction between oppositely charged ions and stabilizes the compound. A molecular formula is the number and types of atoms composing a molecule. An example is carbonic acid, which with 2 hydrogen atoms, 1 carbon atom, and 3 oxygen atoms. You can see an example of a molecular formula directly below the word glucose in the first image. Covalent bonds are formed when 2 atoms share electrons to achieve stability and have a complete outer shell. This sharing creates a strong connection between the atoms. A molecule is a structure formed by atoms joined through covalent bonds. They can be composed of the same or different elements. Molecular compounds are molecules made up of 2 or more different elements chemically bonded. An example is water or H2O. The molecular formula indicates the number and types of atoms in a molecule. The structural formula shows the spatial arrangement and bonding between atoms in a molecule. This helps to visualize how atoms are connected. Isomers are molecules that have the same molecular formula, but different arrangements of atoms in space. The result is different. It results in different properties. An example would be glucose, galactose, and fructose. All have the same chemical formula, however, have different chemical structure. Covalent bonds are formed when atoms share electrons to achieve chemical stability. It happens when both atoms need electrons to complete their outer shell. This can be achieved via the octet or duet rule. The number of covalent bonds is determined by how many electrons an atom needs to fill its outer shell. Types of covalent bonds are based on electron pairs shared. A single bond is sharing of one pair of electrons. A double bond is sharing of two pairs of electrons, and a triple bond is the sharing of three pairs of electrons. Later in this chapter, we will see examples of molecules that have numerous carbon atoms bonded together within their chemical structure. The arrangement of the carbon atoms is referred to as the molecule's carbon skeleton. It forms the backbone of many organic molecules. Three common carbon skeleton arrangements are straight chain, a branch chain, and a ring. Non-polar covalent bonds occur between two atoms of the same type. Both atoms have equal attraction for electrons, so electrons are shared equally. Polar covalent bonds are bonds that occur between two different types of atoms with different chemical structures and differ in their attraction for electrons. The result is unequal sharing of electrons, creating partial positive and negative charges on the atom. Electronegativity is the ability of an atom to attract electrons towards itself when it's part of a chemical bond. Non-polar molecules contain mostly non-polar covalent bonds. Electrons are shared equally between the atoms. An example is oxygen or O2. Polar molecules are molecules that have slightly positive end and a slightly negative end due to an uneven distribution of electrons. They contain more polar covalent bonds. Electrons are shared unequally, leading to partial charges. An example is water, H2O. Amphipathic molecules contain both nonpolar and polar regions within the same molecule. One part of the molecule is nonpolar and the other is polar. An example is phospholipids in a cell membrane. Intermolecular attractions are weak attractions between separate molecules. It helps to determine physical properties like boiling and melting points. A hydrogen bond is a special type of intermolecular attraction and occurs between a partially positive hydrogen atom in one polar molecule and a partially negative atom in another polar molecule. Hydrophobic interactions occur when nonpolar molecules are placed in water or a polar solvent. Nonpolar molecules aggregate together to minimize their contact with water, leading to clustering. This is important in folding of proteins and formation of cell membranes. Hydrophobic literally means water-fearing. Intramolecular attractions happen inside a molecule. They help a molecule keep its shape, especially large ones like DNA or proteins. They link them as internal glue that holds the 3D shape together. Organic molecules are molecules that contain carbon or are or were part of living organisms. Examples include carbohydrates, proteins, lipids, and nucleic acids. Molecules without carbon, except some simple carbon-containing compounds like CO2, are classified as inorganic molecules. Water, or H2O, is an inorganic polar molecule made of one oxygen atom covalently bonded to two hydrogen atoms. Polarity arises because oxygen pulls electrons more strongly than hydrogen, resulting in an unequal sharing of electrons. This polarity gives water many unique properties vital for life. The phases of water include gas, which is water vapor, liquid, which is water, the common form we encounter, and solid, which is ice. Functions of water as a liquid include transportation of substances like nutrients, gases, and waste throughout an organism, lubrication of joints and tissues, cushioning of organs and tissues, and excreting waste through urine and sweat. Key properties of water include cohesion, which is the attraction between water molecules due to hydrogen bonding, causing them to stick together. Surface tension is the inward pulling of cohesive forces at the water's surface, allowing things like insects to walk on water. And adhesion, or the attraction between water molecules and other substances which help water stick to surfaces. Also, thermal properties. Temperature measures the kinetic energy or random motion of atoms or molecules in a substance. Specific heat is the energy needed to raise the temperature of 1 gram of a substance by 1 degree Celsius. Water has a high specific heat, so it resists temperature changes, stabilizing environments. And heat vaporization. This is the energy required to change 1 gram of a liquid into gas. Water's high heat of vaporization means it requires a lot of energy to evaporate, aiding in cooling mechanisms like sweat. A solvent is a liquid that dissolves one or more substances. Those substances are known as solutes. Solutes are substances that are dissolved in a solvent. Water is considered a universal solvent because most substances dissolve in it. Substances that dissolve in water are typically polar molecules and ions. These interact favorably with water molecules and disperse in water. Such substances are called hydrophilic or water-loving. Substances that do not dissolve in water are usually nonpolar molecules. They do not interact well with water and do not dissolve. Such substances are hydrophobic or water-fearing. Substances that partially dissolve in water are amphipathic molecules that contain both polar and nonpolar regions. They partially dissolve, the polar parts interact with water, and the nonpolar parts avoid it. Water dissociates into equal amounts of positively charged hydrogen atoms and negatively charged hydroxide ions. This balance means that water has no net charge and is neutral with a pH of about 7. An acid is a substance that dissociates in water to produce hydrogen ions, also called protons, and anions, which are negatively charged ions. This increases the concentration of free hydrogen ions in a solution. A proton donor is another term for an acid, since the hydrogen ion is essentially a proton that acids donate. Strong acids dissociate completely or nearly completely in water. They produce more hydrogen ions than weak acids. And examples are hydrochloric acid and sulfuric acid. Weak acids dissociate partially in water and produce fewer hydrogen ions compared to strong acids. An acidic solution forms when acid is added to water, increasing free hydrogen concentration, and has a pH less than 7. A base is a substance that accepts hydrogen ions, or protons, when added to a solution, also called a proton acceptor. Strong bases dissociate completely or nearly completely in water and bind more hydrogen ions, reducing free hydrogen concentration significantly. An example is sodium hydroxide. Weak bases partially dissociate and bind fewer hydrogen ions, which leaves more free hydrogen ions in a solution compared to strong bases. An example is ammonia. A basic solution is formed when bases are added to water, decreasing free hydrogen concentration. Also has a pH greater than 7. pH, or potential of hydrogen, is a measure of the relative concentration of hydrogen ions in a solution. It is expressed as a number ranging from 0 to 14. A pH of 7 is neutral, such as pure water. A pH less than 7 is acidic, with higher concentrations of hydrogen ions than neutral. A pH greater than 7 is basic or alkaline, with lower concentration of hydrogen ions than neutral. Neutralization is the process where an acidic or basic solution is made neutral, usually by adding a base to an acid or an acid to a base. Buffers are molecules or systems that resist changes in pH when acids or bases are added. They can be a single molecule or a combination of molecules and help to maintain a stable pH in biologic systems, which is critical for proper function. The information on this slide is important to read through, however, you do not need to know this. Biological macromolecules are large organic molecules synthesized by the human body. They always contain carbon, hydrogen, and oxygen. Some may also contain nitrogen, phosphorus, or sulfur. The carbon component of biological macromolecules may simply be an individual carbon atom or numerous carbon atoms arranged in a carbon skeleton as a chain, branch, or ring. Hydrocarbons are molecules with a carbon atom or carbon skeleton bonded only to hydrogen atoms. They are the basic building blocks in many organic molecules. Polymers are molecules made up of repeating subunits called monomers. Monomers are the individual subunits of a polymer. Each monomer is either identical or similar in chemical structure. A dimer is two monomers bonded together. The process associated with the synthesis and breakdown of complex biological macromolecules is known as hydration synthesis or hydrolysis. Hydration synthesis or condensation reaction is a chemical reaction where water is formed as two monomers bond to create a complex molecules. This builds polymers by removing water. Hydrolysis is the chemical reaction where water is used to break down a complex molecule into smaller subunits or monomers. Lipids are a diverse group of fatty, water-insoluble, or hydrophobic molecules. Triglycerides, phospholipids, steroids, and eicosanoids are the four primary classes. They are the only category of biological macromolecules that are not polymers because they are not formed from repeating monomers. They function as stored energy, components of cellular membranes, and hormones. Triglycerides are the most common lipids in living organisms. They serve as long-term energy storage in adipose connective tissue and provide structural support, cushioning, and insulation. They are composed of one glycerol molecule and 3 fatty acid chains. Triglyceride molecules form by the process of dehydration synthesis. Fatty acids vary in length, commonly ranging in number from 14 to 20 carbons, and may differ in the number and position of double bonds between the carbons in the chain. The types include saturated, where there are no double bonds, and every carbon is fully bonded with hydrogen atoms, unsaturated, which contains one double bond, and polyunsaturated, which contains 2 or more double bonds. Lipogenesis occurs when there is an excess of nutrients. Fatty acids are bound to glycerol via dehydration synthesis to form triglycerides stored in adipose tissue. Lipolysis is a hydrolysis reaction breaking down triglycerides in adipose tissue. This releases fatty acids and glycerol into the blood for energy and nutrients as needed. Phospholipids are the primary components of cell membranes. They are amphipathic molecules that contain both a polar and nonpolar region. They act as chemical barriers controlling what enters and exits the cell. They are similar to triglycerides, but with a key difference. Instead of 3 fatty acids, 1 fatty acid is replaced by a polar phosphate group with various organic groups attached. Phospholipids have a hydrophilic or polar head that is composed of glycerol, phosphate, and organic groups. It is polar and water soluble and interacts with aqueous environments inside and outside the cell. Hydrophobic, nonpolar tails, is the other region of a phospholipid and is made of 2 fatty acid chains attached to glycerol. They are nonpolar and water insoluble, and they avoid water and face inward in membranes. Steroids function as some hormones in the body. They are composed mainly of hydrocarbons arranged in distinct multi-ringed structure. They are characterized by 4 fused carbon rings, with 3 rings having 6 carbon atoms each and 1 ring with 5 carbon atoms. Steroids include cholesterol, steroid hormones such as testosterone and estrogen, and bile salts. Eicosanoids are locally acting hormones that are derived from modified 20-carbon fatty acids that are obtained from phospholipids of plasma membranes. 4 major classes include prostaglandins, prostacyclins, thromboxans, and leukotrienes. Their primary function is in the inflammatory response of the immune system and communication with the nervous system. Other lipids include glycolipids, which are lipid molecules with an attached carbohydrate group. They are primarily found in plasma membranes, contributing to cell recognition and signaling. Carbohydrates are hydrated carbon. The number of carbon atoms typically ranges from 3 to 7. Monosaccharides are the least complex carbohydrate. They are simple sugar monomers and least complex of the carbohydrates and contain 3 to 7 carbon atoms. An example is glucose, which is a 6-carbon sugar and the most common monosaccharide. Disaccharides are dimers formed from 2 monosaccharides bound together. Examples include sucrose, lactose, and maltose. Polysaccharides are long chains made from many monosaccharides linked together. Examples include starch, cellulose, and glycogen. Glucose is a 6-carbon carbohydrate and the most common monosaccharide. It is the primary energy source. Glycogen helps to maintain homeostasis and ensure a continual, adequate energy supply following a meal. The liver and skeletal muscle tissues absorb the excess glucose from the blood and then bind the glucose monomers together to form a polysaccharide called glycogen by a process called glycogenesis. Glycogenesis is the formation of glycogen from glucose that was previously stored. Glycogenolysis is the breakdown of glycogen into glucose, or energy release. And gluconeogenesis is the formation of glucose from non-carbohydrate sources, such as amino acids. Other important carbohydrates include ribose, which is the sugar in RNA, deoxyribose, which is the sugar in DNA, and glycosaminoglycans, or GAGs, which are long polysaccharide chains and important in connective tissue. Proteoglycans are GAGs linked to proteins and the major component of the extracellular matrix. A nucleotide is the building blocks for DNA and RNA. It is composed of three parts: a nitrogenous base that can either be adenine, thymine, cytosine, guanine, or uracil, a phosphate group, and a sugar, which is deoxyribose in DNA and ribose in RNA. A phosphodiester bond is a covalent bond linking the phosphate group of one nucleotide to the sugar of an adjacent nucleotide. This forms the backbone of nucleic acid strands. Deoxyribonucleic acid, also known as DNA, is a double-stranded nucleic acid and stores genetic information. Ribonucleic acid, or RNA, is a single-stranded nucleic acid and is found in the cell nucleus and cytoplasm and plays key roles in protein synthesis. Another important nucleotide is adenosine triphosphate, also known as ATP, which is composed of the nitrogenous base adenine, a ribose sugar, and three phosphate groups covalently bonded. ATP acts as the central energy carrier molecule in cells, transferring chemical energy for cellular processes. Proteins are polymers made up of one or more linear strands of amino acid monomers linked together. An amino acid is the monomer unit of proteins and contains a central carbon, an amino group, a carboxyl group, and a unique R group, which is a side chain that determines its properties. The R, or remainder group, is the variable side chain attached to the amino acid's central carbon and determines the amino acid's chemical nature. Example would be polar, nonpolar, acidic, or basic. A peptide bond is a covalent bond formed between the carboxyl group of one amino acid and the amino group of another. This links amino acids together in a chain. The N-terminal is the start of the polypeptide chain, where the free amino acid group is located. The C-terminal is the end of the polypeptide chain, where the free carboxyl group is located. Oligopeptide is a short chain of 2 to 20 amino acids linked by peptide bonds. A polypeptide, or protein, is a longer chain of more than 20 amino acids. When folded into a functional shape, it's called a protein. Glycoproteins are proteins with carbohydrate groups attached and are important in cell recognition and signaling. Amino acids are classified into nonpolar amino acids and polar amino acids, also charged amino acids, and amino acids with special functions. Nonpolar amino acids have hydrophobic side chains that do not interact well with water, typically found inside proteins away from aqueous environments. Polar amino acids have hydrophilic side chains that can form hydrogen bonds with water, often located on the surface of proteins interacting with the aqueous environment. Charged amino acids have side chains that carry a positive or negative charge at physiologic pH. They are involved in ionic bonds and active sites of enzymes. They are positively charged, meaning they are basic, or negatively charged, meaning acidic. Amino acids with special functions have unique roles due to their chemical properties. Disulfide bonds are covalent bonds formed between the sulfur atoms of two cysteine residues and help stabilize the three-dimensional structure of proteins by creating strong links between different parts of the polypeptide chain or between separate chains.